keynote speaker. He's a professor here at Karolinska Institutet and head of the clinical Proteomics platform at Karolinska University Hospitals and Science for Life laboratory here in Stockholm. Today, he will talk about how to make medicine more precise, effective, and personalized. Please welcome Janne Lehtiö. Good morning, Professor. I will just give the stage to you, and then we will have time for questions through the event app. I leave it to you.
Thank you very much, and thanks for the great opportunity to open this fantastically timely event and theme. So I'm going to talk about precision medicine and diagnostics and how the digitalization is impacting that area. So trying to go from the diagnostics-based medication. So when we have a lung cancer or when we have asthma or Alzheimer's, we are thinking that's a diagnosis. We will medicate that with one type of medications. But we're moving into a medication that will look at the molecular traits and try to understand the individual disease, and by that way start tailoring the medication. So precision medicine is always related to the treatment. Unfortunately, our current medication, diagnosis-based medication is quite unprecise. Here you see some blockbuster drugs, and you can see the red patients are the ones that don't really get benefit of that drug. And the worst case, negative side effects, toxic effects of the drug. And the blue ones are the ones that the drug is really effective on. And this is diagnosis-based medication, how it looks like in many cases today. So if we can start segmenting and understanding the disease and this disease group and tailoring the medication, that's where we need to go. So how do we do that? I'm going to use cancer as an example because that's my own research field and we're working on that. And here is one example what is happening in this precision medicine paradigm, exemplified by lung cancer. So in 1990s we considered lung cancer as a one disease, and then by looking at the microscope, we see that there is a different patterns. So that's divided into a different lung cancer entities, small cell, non-small cell, and further into division. And then about 15 years ago, the genome medicine era started, where we start looking at the molecular traits. And that's a trajectory that we are adding now in more information to the molecular image, to the histopathological image, and now proteome, metabolome, whole body imaging into this. And that way we segment lung cancer into multiple disease entities. What type of lung cancer it is, what are the drivers of that? And why does this digitalization matter to all of you? Because these diseases affect all of us. And this trajectory of diagnostic data generates lots of data. So let me start by introducing you to Karin. Karin got lung cancer in 2014, and that's a very severe diagnosis. Unfortunately, her disease was metastasized to several places in the body, and at that time the treatment outcome and the outcome was very poor, approximately 2 years, and many times even less. But this is where we had started to disseminate the 20-40 years of research into genome medicine, where we have these medications. And we already had a clinical trial at the Karolinska with a particular mutation that Karin had in her tumor. So she was enrolled in this trial. This is one important message, that we need clinical research and clinical trials, not only for the research and knowledge generation, but it will give early access to the new innovative treatments. That medication worked really, really well. and the tumors shrink. And unfortunately, Karin has got several relapses, but because of this medical development and data generation, there was new medications that were again effective. There was new precision radiotherapy using the imaging combined with steering. Today, I met Karin last summer in the Almedalen, where we were talking to politicians how important it is to put money on the precision medicine and diagnostics. She's feeling well, she's having 2 tablets per day. She was telling about her holiday plans and grandchildren and so on. And this is not a miracle. This is systematic precision medicine work and diagnostics development that leads to this type of situations, and we need to gather the brain power that we have in the digital community also in this type of work. And that's why I'm here. I'm inviting you all to contribute to precision medicine with your brains and with your tools. This illustrates that we have not solved the problem. We have innovative new drugs like the immunotherapies in cancer. This is immunotherapies that are very effective in lung cancer for the patient populations that don't have these particular mutations, but they still do not cure the patients. They prolong the life, so we need more. And why do they do that? Is that we need combination treatments. What happens is that these diseases is like an evolution in a mini scale. They find ways to go around the medication, like the water in the waterfall, if you dump that in one place. So what we do need is better mapping. So we need combination treatments. We know how to do this. This is an example of HIV treatment that we sort of solved as a scientific community together in the precision medicine already in 1990s by combining multiple drugs, turning the HIV from the death sentence to a chronic disease, and has been improved. So this is what we need. The cancer challenge is more complicated because it's much more heterogeneous than one virus. So what we do need is better digital maps of what is going on. What is going on in the cancer? So this is illustrated by the same waterfall in different projections. How do we tailor the treatment combinations when we have more information, when we are on the map, when we understand the system better? And this is where we are heading at. So we will collect all these data, medical records, radiologicals, histopathological data, genomics, proteomics, spatial biology, put that into a context of treatment outcome, prognosis, how is it going for the disease, what type of response did we obtain. So we need to build this data together with the clinical outcomes and form these iterative learning loops. So what are the collaborative opportunities and conflicts here? So we have a few dilemmas here. Why are we not curing all the cancers already? And I'm telling you a few aspects of this. One is that the data that you saw in the previous slide is very complex. We have lots of multiple different data types that are interrelated, and you need to understand a lot about the data, how to wave it together. The other problem is that we cannot really use the AI tools because the data is not freely available in that massive scale related to the things that matter, outcome and so on. The other thing is exemplified in this picture, where we have mapped the stakeholders by building the Precision Medicine Center Karolinska, and this is just a zoom in of a little part of the stakeholder map. There is lots of stakeholders, there is lots of people that want to work on this. There is companies, there is academic things, there's hospitals, there's healthcare providers, there are patients, patient organizations. how do we actually synergize in a best way? How do we collaborate in this stakeholder landscape? There is also only one stakeholder, and that is the patient. Can we do something that really matters for the patient? And that one stakeholder perspective actually lifts another issue, that the data is very sensitive. We have a conflict of the personal integrity and benefiting and learning from the data to cure and to find the right treatment. So how do we approach this? I'm going to then say how do we build these collaborative networks and what are the structures here. So one way to do this is to systematize the problem from academia and industrial research to healthcare. So first you need to bring in these technologies and this data that is generated by these technologies to innovative clinical trials, because that's our systematic way to develop diagnostics and treatments. And then you need to have a system to scale these innovative clinical trials to a population-based routine use. So both these needs quite much effort, and then you need to be able to use that healthcare data for the innovative loop to start developing things that didn't work in the first round. So one of the things that we are doing locally is Precision Medicine Center Karolinska, where we have collected the stakeholders into a center that is driving this development at the hospital, the local universities, and of course the Karolinska Institute as the biggest medical university has lots of involvement on this. Also Science for Life laboratory that I'm going to talk a little bit about. And then we have, out of that work, we have developed different sub areas where we work on the legal aspects and also, but one of the things that we are working now is a precision medicine hub, where we want to bring these technologies close to the hospital, start generating this data in a large scale, and then start connecting the people that can AI and data analysis into this scheme. And then start using that in the innovative clinical trials, parallel developing how do we move those technologies and data into the routine use at the Karolinska Hospital and other places. One of the assets that we have is Science for Life laboratory that we're very happy in Sweden. I'm of course very biased because I'm scientific director also at the Science for Life laboratory, but why I think and why many others think that is so great is that we have connected cutting-edge research community with well-financed technology platforms that are continuously developing these cutting-edge technologies, molecular imaging, proteomics, genomics, and so on. And now we were very lucky also to add completely new data dimension with a data-driven life science program that was financed by Knut and Alice Wallenberg Foundation. And by the same time, we have broadened this into a truly national network of all the Swedish major universities, and we are building network with the university hospitals to bring in technology and data-driven precision medicine and life science into not only on precision medicine, but we are working also on environment and biodiversity and so on. These are the technology platforms. All these platforms can generate very, very valuable data for individual patient decisions if we can harness these platforms to generate that data. And this is what we have been doing in the last 2 years to build a precision medicine team to actually work with the healthcare and universities developing and integrating these technologies. We started by genomics 10 years ago, and that's actually moved already in many places to the hospitals by the technology backbone that we started. But we have 9 other platforms that we could really contribute to. The data-driven life science program, which is driven by SciLife Lab, financed by Knut and Alice Wallenberg Foundation, is really a program to bring in talent into the life science field. And one of the important areas is precision medicine and diagnostics. So we are recruiting top names around the world, young innovative researchers here to work with us on these themes. We're also building in sustainable structures to warehouse the data, to tap that into the data resources. So this is an example of one of our recruits, DDLS fellows, Jakob Vogel, who is working on Alzheimer's, looking at the brain images and trying to structure that, and actually finding different trajectories, how the disease, different patient subgroups, and then using the SciLife Lab platforms to generate molecular data to understand what are these different disease entities that we can decipher from the brain images. And then trying to combine these two data sets to understand the disease course and the molecular profiles to actually stratify patients for clinical trials and to get more precision medicine drugs into right patient groups to be tested. So this is an example of these young researchers that we bring in to work with us. So this is the 4 areas what we are working on: precision medicine diagnostics, we have cell and molecular biology, epidemiology and infection biology, and evolution and biodiversity. I'm area lead on the precision medicine and diagnostics, that's why I'm preaching to you about that. But there we are trying to couple the technologies and data science to university hospitals, to universities, and also bring in collaboration forms with the industry. We have industry PhD programs and so on. Locally at the Karolinska Institute, we have DDLS Finance Data Science Node that is bringing in people to work with these structures and systems, data storage and so on, and knowledgeable people, data stewards, to work with people from different stakeholders, from the healthcare, from industry, to bring these collaborative networks. We are also very happy about the fact that the Wallenberg Foundation, together with the industry, is financing another really big project in Sweden, which is WASP, AI and autonomous systems and softwares on the industry side. And we have bridging financing to tap into the brains and knowledge and systems there. And we are also using the same supercomputing resources, and we are connecting the life science with the research community that is working on industry development on AI and systems. This sits in the European context then of European health data space and digital Europe developments, where health is a very important sector. And we have been very fortunate in Sweden to be part of all of the initiatives on the health domain that have started so far. And Vinnova in Sweden have catalyzed the financing for SciLife Lab to coordinate these digital innovation programs in European scale. So we can tap the Swedish and the Stockholm development all the way to the international context. So I'll turn in the last few minutes back to the cancer and what this type of data can do. So this is a leukemia cohort that we analyzed by generating new, innovative, interesting data layers: genomics, transcriptomics, proteomics, methylation data, drug ex vivo data, and clinical history. And by looking at the molecular phenotype, the proteome that we actually target with the drugs, we could see that it structures these leukemia cases in 6 different groups. And when we connected the clinical history data and the clinical outcome data, we could see that these groups are really prognostic. If you assign patient to one of these groups, you can then immediately tell how is the likelihood that the disease trajectory is for that patient. So that means that this molecular information is telling us something about the disease and the disease course. So then we went back to the molecular information and started to look. If we connect the genomics and proteomics information, we started to see frequently reoccurring genomic events and how they impact the proteome. And is there proteins that you can actually target? And by analyzing this, we could realize that there is certain genomic changes that we can couple to certain treatments that were not known before. In this case, B-cell receptor inhibition. So we took retrospective large clinical trial material, reanalyzed that material, and showed that actually we can couple new genomic treatment. So we can find new patients that benefit on B-cell receptor inhibitory treatment and do much better, just by layering these data points. So what happens there is that if you look at the genotype separately, it tells you one thing. If you look at the molecular phenotype, you can see the joining areas there and connect treatments to there. So this is what I was telling you about the waterfall. By creating this map, we understand what treatments on what patient populations we can connect and how do we connect this. So this we have been developing digital tools, and this is collecting cancer genomics data in 7 centers in Europe. Here to Stockholm, we annotate and analyze that data and make report to the clinical team. And that report is discussed in a pan-European molecular tumor board between the 7 leading cancer centers. So if there is a difficult case, we can do the analysis, we can make the molecular report, and then we can tap into the brains of all the top clinicians in these 7 centers. So once a week, we are looking at these patient cases catalyzed by this digital tool that everybody has learned to use. And this is connecting people in where it really matters. And this is something that we want to do in Sweden with a Testbed Sweden clinical trial. So we are starting this type of thing, collaboration also across the Sweden in cancer side, and adapting the same digital tool. But this time we hope to add much more technologies that we have available at the SciLife lab to generate even better data. So we want to build this iterative loop where we do experimental research and we feed that into the hospital, both the data and the technologies and knowledge, and then start using these in innovative clinical trials, bring the data back and the new research questions back to the academia. And with that, I'll start by showing this picture. It's actually a tube map of Paris with a peak hour traffic as a heat map under. Just by connecting these 2 data sets, you will get much more information. So that's what we need to start doing, and to understand what are the data layers that we connect to do precision medicine, to make that a reality. So thank you for your attention.
Let's move over here for questions.